A Great Dane will probably be dead before its 10th birthday. An elephant lives 70 years. A bowhead whale lives over 200. Across many species, bigger means longer lived. Dogs broke that rule. Irish Wolfhound, one of the tallest dog breeds in the world, median lifespan 6 to 7 years. Chihuahua, 15 to 17. We did this through breeding. Here is the biology of how. It starts with a single hormone, IGF-1, insulin-like growth factor 1. IGF-1 tells the body how big to grow. In 2007, Nathan Sutter and colleagues published a study in Science showing that a single gene, IGF-1, has an outsized effect on size variation in dogs, particularly in distinguishing small breeds from large ones. Small breeds tend to carry IGF-1 variants associated with lower growth factor activity. Large breeds tend to carry variants associated with higher activity. IGF-1 is not the only factor, but it is one of the most important. It does two things simultaneously, and both of them shorten a dog's life. The first, high IGF-1 drives rapid growth. A Chihuahua reaches full size in 6 to 8 months. A Great Dane takes 18 to 24 months, continuously building bone, muscle, and organ mass. Growth requires cell division. Every cell division carries a small chance of a copying error, a mutation. More growth means more divisions. More divisions means more accumulated errors. In 2013, Cornelia Kraus and her team at the University of Göttingen published a study in the American Naturalist analyzing records from 74 breeds and more than 56,000 individual dogs. Their finding, for every additional 2 kilograms of body weight, a dog's expected lifespan decreases by approximately 1 month. The trend held across breeds, sexes, and countries. Meanwhile, the second thing IGF-1 does is activate signaling pathways, including one called mTOR, that suppress autophagy. Autophagy is the body's cellular cleanup system. It removes damaged proteins, breaks down dysfunctional components, and catches mutated cells before they can replicate out of control. When IGF-1 is high, autophagy activity tends to decrease. Large dogs are likely accumulating more cellular errors from accelerated growth. At the same time, their cleanup system may be running at reduced capacity. More errors, less cleanup, both, in theory, driven by the same hormone. In many studies, cancer is the leading cause of death in adult dogs, accounting for roughly 40 to 60% of deaths, depending on the dataset. The Morris Animal Foundation runs the Golden Retriever Lifetime Study, one of the largest health studies ever conducted on a single dog breed, tracking more than 3,000 dogs from birth to death. Prior data from the study suggest that more than 60% of Golden Retrievers will develop cancer during their lifetime. It is the leading cause of death in the breed. Large dogs do not get cancer more often primarily because of bad luck. The elevated rate is likely linked, at least in part, to the IGF-1-driven dynamics described above: more cell divisions, reduced autophagy activity, errors accumulating faster than the body can clear them. In many wild species, larger individuals tend to live longer than smaller ones. Why not dogs? The answer is the speed of selective breeding. In wild animals, large body size evolves over thousands of generations. The metabolic systems, the repair mechanisms, the cellular maintenance pathways, they all evolve alongside the body, in step, over the same timescale. Dogs were selectively bred to extreme sizes within a few hundred years. In evolutionary time, that is nearly instantaneous. The body grew, the maintenance systems did not catch up. In wild wolves, the body size range is narrow enough that this kind of extreme size-lifespan trade-off simply never had the conditions to emerge. wolves were never pushed to the size extremes that selective breeding created. A Great Dane's body was built in centuries. Evolution would have needed millennia. There is active research trying to change this. The Dog Aging Project at the University of Washington is running a clinical trial testing rapamycin, an mTOR inhibitor that, among other effects, can promote autophagy activity. Early results are cautiously promising. For now, there is no fix. One leading explanation looks like this. IGF-1 drove the growth. Growth drove the cell divisions. Cell divisions accumulated errors. Reduced autophagy let those errors build. Many of those errors contributed to cancer. This was already in motion the moment that dog was born. It is a biological consequence written into the genome. Humans reshaped a species faster than evolution could follow. If you have a large dog, or if you have lost one, tell me about them in the comments. And if this changed how you think, subscribe. There will be more of this. One mechanism, one chain at a time.